Radiata, Glacier, and also recently I had a chance to work on the fungi associated with shallow hydrothermal vents in Taiwan. I'm going to give you a brief outlook of the technological application of marine fungi. Now, I think I've said it yesterday, or some of you were there yesterday, but I just want to give you a brief, because some of you were not there yesterday. I give you a brief introduction to marine fungi, because you might wonder why. What is marine fungus? Why is it different from terrestrial fungus? Basically, we have three criteria for defining a marine fungus. Basically, the first criterion is if they are able to grow and sporulate in the marine environment, we call them marine fungi. So this is a fruiting body that you can find on the organic substrate in the marine environment. Basically, it's just like a mushroom growing on a piece of wood. Okay. The second criteria, if you can observe a symbiotic relationship between a fungus and other organisms in the marine environment, we can call them marine fungus. For example, you can see a lot of marine lichens. Okay, you can see them. So they are marine fungi. And also, there's another example that I introduced yesterday is a snail and a fungus forming a symbiotic relationship or commensalistic relationship on the marsh grass Spartina alterniflora. And finally, the third criterion is if you find fungus that metabolically active and they can adapt to or involve epigenetic level, you can call them marine fungi. Basically, if you can find a fungus that is phylogenetically related to the lineages of marine fungi, you can call them marine fungi. Or nowadays, you use a lot of molecular techniques to look for active population of fungi in the marine environment. Say, for example, if you use RNA, then you know that those are the fungi that are active, and they are actually marine. So they're different from the terrestrial fungi that we know about. Now, this graph shows you the number of marine fungi in different decades. Okay, actually, the first marine fungus was described back in 1840, so around that time. And it's not until 1944 when an American called David Linder initiated a study of marine fungi in coastal water in Massachusetts. And then after that, this study triggered quite a significant interest in the study of marine fungi. So you can see that after 1944, there has been a lot of studies describing marine fungi on various substrata. Okay. At the early days, people looked on the fungi associated with wood or seaweeds, and later on, people look at the grass, the marsh grass, and also the fungi that are forming fruiting bodies on sand grains. And in the last 20 to 30 years, people have started to look at fungi associated with juncuses and other marsh grass, and also the nipa, which is a palm that in the tropical areas. Now, for the study of fungi, normally there are various techniques. The most common technique is to identify fruiting bodies on the substrates. Now, normally a lot of people in fungi, they will form fruiting bodies on the substrates. In this case, it's a piece of wood. Okay, sometimes they're immersed, sometimes they are exposed on the surface of wood. And these two are the deep sea fungi we collected 3,000 meters down in the sea bottom of near South China Sea. Another technique that we use is for isolation technique. Okay, basically we can put the substrates on a nutritious medium, and then the fungi will grow from the substrate, and then you can isolate these fungi as pure cultures. And another method, say for example, if you're working on seawater or sediment, you can do serial dilution and then plate them on nutritious media, and also you can isolate the fungi growing on this nutritious media as pure culture. And a lot of the time when you identify these pure culture, it's based on the identification of the internal transcript space region of the ribosomal DNA. So after using all these techniques, over the years, we have around 1,100 species of marine fungi. Okay. Most of them, they are the Ascomycota. It's about 80-something%. Okay. Now I know that some of you were there yesterday, so you know what an Ascomycota. But for those who don't know, Ascomycota include the fungi such as truffle. Okay, the one that is very expensive fungus. And another is 冬虫夏草, winter worm sungrass. I think in English it's called that. Okay, normally you can find it in Tibet. Okay, they are all included in the Ascomycota. And there are about a few percent of marine fungi that belong to Basidiomycota, which is the group that you know the best, which is the mushroom group. Okay. And for the rest, there are other groups like Psychomycota and also zoosporic organisms, and they only comprise part of the total number of marine fungi. Now, briefly look at the main groups. The marine Basidiomycota I told you yesterday, they are the mushroom of the sea. Okay, and there are about 21 species. And most of the mushroom in the sea, they are very susceptible to salinity. So the places you normally find them are the high tide area of mangrove. Okay, I don't think you know. Well, some of you got to know mangrove, but mangrove has only subtropical and tropical distribution. Okay, and for the mushroom in the sea, normally they have very reduced fruiting body. It's not as big. You know, in the terrestrial environment, you have very big fruiting bodies. You know, the foliates, they can be as big as this. Okay, but in the sea, they are not very big. They're about maybe, you can see them, but if you want to look at them carefully, you need to use a microscope. Okay, so they're comparatively tiny. And in the marine environment, there are quite a number of yeast. There are about 75 species of yeast in the marine environment that belong to the Basidiomycota. So the mushroom, there are quite a number of mushrooms in the sea. The main group in the sea is the marine Ascomycota, okay, which means that they form acid, okay, which I told you include 冬虫夏草 and also the truffle. Okay, there are about 800 species, okay, and there are about 130 species of yeast in the marine environment as well. Okay, for this group has been studied quite extensively. Okay, and we know that there are quite a number of morphological adaptation through evolution, such as the spores. When you look at the spores of this marine or aquatic fungi compared with terrestrial fungi, they normally have large oil globule, which help them to float in the water, and also they have appendages to help them to attach to substrata in the sea. Okay. There's another group. Okay, there's marine asexual fungi. So basically include two groups. One is the coelomycetes, which form fruiting bodies on the wood or on the substrates, and others, they produce spores directly from the mycelium. Okay. Now, I told you yesterday, in fungi, the classification is only based on the sexual structure. Okay. For the asexual fungi, they are not formally classified into the classification system of fungi. Then you might ask, why? How can we know where it belongs? Normally, nowadays, we normally sequence these asexual fungi and use phylogenetic analysis to know where they belong. Okay. So, but there are quite a few, 40 to 50 species in the sea. Now, we have been looking at substrates from a wide range of substrates. I've been to different places and collected different materials. There are some. I'll give you a summary of the fungi associated with different substrates. The most studied group of marine fungi are actually on wood. Okay, I was thinking when I was here whether I should collect some wood back to examine, but I don't know very well. But I was told that it's quite far from the sea. I think, right? It's quite far from the sea, and also the water here is not actually very saline. Am I right? They're brackish, more brackish. So I decided no. So, but the wood is the most studied group of marine fungi. Okay. So you can sometimes, if you go out to the seashore, you can find wood trapped between rocks. Sometimes you can find wood buried in the sand. If you go to mangrove, there are quite a lot of wood that you can collect, and a lot of fungi growing on those woods. You don't know them, you don't see them, but actually they're there. Okay. So there are about 300 species of marine fungi being reported on mangrove wood. And I'm not going to talk about the taxonomy, but what you need to know is, if you go to collect wood from different places, say for example from sandy beaches or from rocky shore, the diversity of fungi is different. Okay. Another group that has been studied a lot is the salt marsh grasses. Okay, there has been a lot of study on Juncus romarianus in the US, and a lot of study on Spartina in Europe. Okay, and in more tropical area, there has been a lot of study on Phragmites. Okay, Phragmites, they're everywhere. Okay, they can be terrestrial, lower. Okay, terrestrial, they can be freshwater, they can be marine water, and there is a high diversity of fungi associated with these grasses. And recently, I have been to the south of Taiwan, and my main target wasn't to collect Phragmites, but I did collect some Phragmites back. And there are quite a number of new species on this grass. So one of them is, I described this species from Phragmites. Okay. So whenever you have the chance to look at fungi associated with grass, there is a high chance that you're going to get new species. Okay. Another group that has been studying a lot of seagrasses. I'm sure you have seagrasses here. Okay, especially if you look for phosphochytrids, you can try to isolate phosphochytrids from seagrasses as well, because a lot of these phosphochytrids, they are associated with plant, okay, or macroalgae. Okay, so there has been a lot of study. We don't know the number of species of marine fungi associated with seagrasses, but we know that the majority of the fungi belong to the Ascomycota, and some of them belong to the Ascomycota. And the dominant species you might know are the Aspergillus, Penicillium, Acrylonium, and also Plectosporium. All these species are very common in the geospheric environment. Of course, fungi has been studied on marine animals, told you yesterday on corals and on sponges, and you might ask, why do we study fungi on these animals? Because recently we have found, especially natural product chemists, they have found quite a number of interesting structure from microorganisms associated with these animals. That's why recently there has been a lot of study on this group. And again, the fungi that you found on this animal are common terrestrial genus. Okay, like Aspergillus, Penicillium. You can easily isolate them from these animals. But of course, there are a lot of fungi associated with macroalgae. Okay, this is fucus, and I'm sure you have fucus here as well. Okay, this is a brown alga. Okay, there has been over 100 species described from macroalgae, and you can find them either they form parasitic, symbiotic, or saprobic relationships with other substrates. I think a lot of people study fungi and algae because they cause disease of the algae. Okay, I told you yesterday, some of them they can destroy the algae, just like this one. They can form a lesion on the algae. Okay, they form fruiting bodies on the algae. And a Chinese colleague, he basically summarized the fungi that can cause diseases of algae, and he has summarized there are 31 species and 1 asexual fungus that can cause disease of algae. And I told you yesterday, it's not a fungus, but a lower marine fungus can cause disease of the papyrus, which is a red alga that we use in China, in Japan, and in Korea to produce the dry seaweed. Okay, I told you yesterday, if you have eaten sushi, then you definitely have eaten papyrus. And this pigment species can cause disease of the algae and kill the algae. So the production was much lower. Okay. I have studied algae recently. This is the alga that I studied Coccoteria Capitulaceae. And in northern Taiwan, I don't know how much you know about the red algae. You know the red algae, they produce the agar. You use agar, right? Okay, the red algae, the agar was actually extracted from the red algae. So in northern Taiwan, we use the red algae to generate, it's not to prepare like a dessert, just like a jelly thing. Okay, it doesn't taste very nice, but the reason why is agar, it has no taste. It's just like gelling agent. Okay, you have to add flavors into the agar, into the dessert. But we normally go and collect a lot of these 我们叫海女. There's some divers, you know, those lady divers. They will collect these red algae at the seashore, okay, to make those desserts. Okay. So, but I have a different reason to study this alga. The reason why is there, although there has been some study on the fungi associated with algae, but they are still under study. Okay, and in northern Taiwan, there are a lot of algae. Okay, so and they're on the shore. There's so much. Okay, compared to wood. So I started to wonder why there's so many healthy algae there. Okay, because in the seawater, if you can imagine, there are quite a lot of viruses, quite a lot of bacteria, quite a lot of archaea, quite a lot of fungi there. Some of them they're pathogenic. Okay, I wonder why they're so healthy. Okay, is it because the microbe associated with this algae, they give this advantageous effect to the algae, such as they produce compound to protect the algae from infection by the microorganisms in the sediment and all the seawater. Okay, that was one of the reasons why I studied the fungi associated with this red algae. And of course, another main reason is my colleague, who is a natural product chemist, is very interested in looking at the natural products of fungi associated with algae. And he has his own reason. The reason why is there has been quite a number of new structure isolated from fungi associated with marine substrata. And as you can see here, there are 20% of the new structure actually isolated from fungi associated with algae. So we initiated this project at a place like this. I don't know how, some of you might know Taiwan very well, but that is the place that we collect. That is the place where my university is situated. So this is the site. Quite nice. Okay, it's a sunny, it's a sunny day, but it's raining. It's not very nice. So basically, we look at the fungi associated with healthy and dead Ka-Lai, and then after collecting the samples, we surface sterilize the algae or wash the algae and plate them on different media. And also, we want to know if there are seasonal variation. I'll give you some insight. If you are studying diversity or ecology or fungi or bacteria, this is there's some information I think that's useful to you, to your research. Okay. So basically, we went to collect, okay, once every 2 months. Okay, so we collected the algae for I think 6 times or 7 times, I don't remember. We collected the healthy algae, which is red. There's some dead ones, okay, which is completely bleached. And then after collection of samples, we separate the healthy and dead Ka-Lai, and we wash them, okay, either with seawater, with ethanol, and sodium hypochlorite. You might wonder why we use. Do you know sodium hypochlorite? It's a bleach that you use to disinfect things. You know, bleach and also ethanol. You might wonder why we want to wash them, because I'm interested in the fungi living inside the alga. Okay, so you have to surface sterilize the fungi. You kill all the fungi on the surface of the alga in order to get the fungi living inside the alga. Remember I said endophyte yesterday, the things that living inside the plant. Okay, So I'm more interested because the endophyte, some people say the endophyte produce compounds to protect the algae. So I want to test this. So that's why I use seawater to wash, or I use ethanol and sodium chloride to disinfect the surface of the algae. And then I play them on different media, the seawater agar, the glucose C6 recaptome agar, and protein ester agar, which meaning that I'm giving them different media to see if they grow. Yesterday I said if you isolate microbes from a substrate, you can normally isolate 1 to 5% of the total microorganism in the sample. The more media that you use, the higher diversity that you're going to get. Okay, so if you only use one medium, you get lower diversity. If you use more media, then you get a higher diversity. That was the reason. Okay, it's because different media will have different nutrients inside the medium. So after washing them, plating them, and then we incubate them, basically they will grow like this. Okay, it looks quite gross, right? But because there are a lot of different fungi, so basically what we do is we cut a piece of the mycelium, okay, from these. This is a piece, and then we will purify it as pure culture. Okay. So what do we do? Because this is the isolation, we have to identify them. Yesterday I told you for the barcoding gene, in order to identify the fungi, you use the barcoding gene because different fungi will have different barcodes, okay, based on the internal transcript basal region of the ribosomal RNA gene. Okay, that's what we did. Okay, now we totally investigated 4,450 alpha thalli, and then we isolated 3,187 strains, and then basically we could not afford to sequence 3,000 strains. Well, basically, the first step we did was to classify them based on morphology of the colonies. And then after grouping them, okay, we sequence the internal transverse base of the RNA to identify them, okay, based on the 97% cutting point, okay, sequence similarity, okay, with the sequences in the NCBI database, okay. So this is the result. Now this graph shows you the isolation frequency. That means the number of thalli with fungi growing out. Okay. Now first of all, we look at the different washing method. This is washed by artificial seawater. This is washed by ethanol. This is washed by sodium chloride. As you can see, more fungi was isolated from the thalli washed with seawater, which is very reasonable because these are very hypochlorite, powerful disinfectants. So it kills a lot of fungi on the surface of the algae. So you get lower number of fungi isolated from the thalli washed with sodium hypochlorite. Okay, and the ethanol is in the middle between the two. Okay. Now, when you look at another way, you look at the media, okay, which is these 3 bars. Okay, you can see that the GYP, which is the glucose media, has a higher number of fungi growing out. The reason why is glucose is a very easily used carbon source. So the fungi like glucose, so they grow quickly. Okay, and then you get a higher diversity. Comparatively, you can see that when you use either seawater agar, which has no nutrient in it, just seawater and agar. Okay, so there's not much nutrient in it. So and the fungi won't like this media. So you can see that less fungi were isolated from this media. Okay. Overall, when you look at the overall diversity, you can see that the majority of them they are ectomatosis. Okay, they are about 80%. There are about 18% that they see the microta, and a lot of them are insects. Okay. Now you might wonder why there are some uncultured and unidentified. I think some of you might have this experience when you sequence the ITS region. When you compare with the NCBI database, there are quite a number of unknown sequences in the NCBI database. Okay, and that's why we have some sequences, but not a lot. About 3 sequences we didn't know what they are. Now, first of all, we look at the diversity of fungi between the dead fungi, which is the inner circle, and the healthy Ka-Lai, which is the outer circle. As you can see, the colors are quite similar. Okay, meaning that they have a similar group of fungi associated with the healthy and dead Ka-Lai. Okay, but the differences are the abundance. Okay, you can see that the same order, the abundance will be different. Say, for example, the dead Ka-Lai you have 33%, the healthy Ka-Lai you have 25%. For this one at 7%, 12%, that's 6%. So they're similar in species richness, but they're different in species abundance. Okay, abundance meaning the number. Okay, richness means the number of species. Okay. I told you I'm interested in to see whether there are differences. There are any endotypes between in this elder. So we compare the diversity between the healthy and dead Ka-Lai. So you can see that there are 70 species that are common between the healthy and dead Ka-Lai. Okay, but there are species that are only isolated from healthy Ka-Lai. There are 7 species only from the dead Ka-Lai. Okay, but the common Ka-Lai, the common fungi in both Ka-Lai, they are the Rhodosporidium, which is the pink yeast, and also the Aspergillus species, and there some Fusarium. Fusarium is a very common species in sediment and seawater, and there's some species that are only isolated from the healthy Ka-Lai. If we look at the different washing methods, okay, there are some species that are common to 3 washing methods. There are 59 species. But there are species that are only isolated from some of the washing methods. Okay. The most, the fungi that interest me are the ones washed with sodium hypochlorite. Okay. The reason why is you can see the hypoxylon in plastics and also in many. These are very common terrestrial fungi. Okay. They infect plant seeds in the terrestrial environment. But they have been repeatedly isolated from the sea. And because these Ka-Lai being washed with sodium hypochlorite, we are quite confident that these fungi are actually living inside the plant. And this group of fungi are known to produce compound that has antibiotic properties. Okay. And this is the difference between using different media. Okay, there are 69 species that are common to PDA, seawater area, and also GYP, but there are also species that are only isolated from individual media. Okay, you might wonder why I tell you all this, and the reason why is I told you earlier. If you are working on diversity of either fungi or bacteria on particular substrates, you have to try to use different methods to investigate the diversity. The more method that you use, the more realistic diversity you're going to get, because you will never know the true diversity on a substrate. You can only try your best, try to use more methods to get to the true diversity. Okay, just try to use more methods. Now, I told you I look at the seasonal variation as well. Okay. Now this is the graph. Okay, shows you this is the abundance. This is the species richness. You can see that during the summer months there is generally higher species richness, which means that it has higher number of different species. And in the summer months they also have more isolates. Okay, than the winter months. Okay, it's not as cold as in Tianjin. Okay, but it's around 10 degrees, 8 to 10 degrees during the winter time. Okay, so it's not so cold. Okay, but you find there's lower species richness and also lower species abundance during the comparatively colder months. Okay. Now we also did a Gray-Curtis similarity index because this method is to compare the fungal community between different months. As you can see that the fungal community in the summer months are more similar, and so as the winter months. Okay, so basically what it tells you is there is a seasonal variation of fungi associated with this. Okay. Now nowadays you have to use molecular method to study diversity. Okay, people will use cloning or use sequencing, but nowadays high throughput sequencing is so cheap. Okay, so people will opt for high throughput sequencing. That's what we did. Okay, we used a nested TCR approach to amplify a part of the gene of the ITs. Okay, to look at the diversity of the fungi. Now, the first set is using these two primers. The second set is using these internal primers. You might ask, why don't we just use one set of primers? Because when you use nested PCR normally, you skew the diversity. Okay, because PCR is a biased tendency. Okay, we have actually sequenced the amplified by these two primers, but the organisms that we have are a lot fungi. So we thought we use an internal primer more specific to fungi to see whether we get more fungal sequences. Okay, that's why we use this nested PCR approach. Now I can tell you that actually these primers are not designed by me. Okay, these are the primers that in the paper they said fungal specific. You will never get fungal specific primers. Okay, so remember, so if you have a project to look at diversity using specific primer, try to use different primers and try to select the best primer for sequencing. Okay. Now we are still analyzing it, but I'll give you. We use Illumina high-sense sequencing. These are some of the results that I'm going to show you just to show you the community. Okay, we're still analyzing the database. As you know, when you do these high throughput sequencing, the database that you use is actually a closed database. It's not an open database, meaning that you are not going to get the most updated sequences in the database. Okay, so you're trying to compare with internal database. Okay, it's not like NCBI. So basically, what we are doing at the moment is we extract all the sequences and we blast each sequence with those against those in the NCBI database. NCBI is more updated, so you have a more updated taxonomy of the sequences that you're going to have. So if you have a chance, I would recommend you to do, but it also depends on the number of OTU that you have operational taxonomic unit that you're going to get. So, but from this graph, you can see that these are the fungal community in different months. As you can see, the different colors represent